All right, so starting with mitosis and the stages of the cell life cycle. So the cell, and I'm like, a cell is born. It doesn't always live forever. And I'm like, especially in certain parts of the body, it wants to divide. So we always have constantly new cells all the time. Now, depending on where the cell is in its life cycle, and I'm like, we've got various phases. If a cell is an interphase, it's just kind of hanging out, maintaining. It's not wanting to divide. It's just hanging out, maintaining. Inter means between. It's like the in-between phase. It's just hanging out. And I'm like, it carries out all of its normal functions, makes ATP, makes proteins, whatever else it wants to do. When the cell decides it needs to divide, we need additional cells, we need replacement cells, that's when it goes into the mitotic phase or the cell division phase. So that by the end of mitosis, you'll have now 2 cells. Now, interphase, that doesn't mean there's nothing going on in interphase. It is the maintaining phase, kind of the hanging out phase. It's in between cell division, but we can get some, this is the part you'll see your nuclear material is called chromatin. Now, chromatin is not coiled up tight. It's this kind of loose DNA floating all around. And because it's so loose and so fine, you don't see anything under our microscopes. It just kind of looks like fuzzy grains. And I'm like, that's when you know it's an interphase. It's just fuzziness. And I'm like, that you see on the microscope, because that chromatin is so thin. Now, because some stuff does happen in interphase, and I'm like, interphase is split up into 3 subphases, a G1, an S, and a G2. And I'm like, the G1, I'm going to scroll back and forth because I just like this big picture. So all of the green, where it hangs out in most of its life, this is all interphase. All of this green, teal, that's all interphase. But things happen. One, it starts to grow. I'm like, it has a new, it was born a new cell, it needs to grow. We may add some few more organelles. It's going to repair itself a little bit. So we have a growth phase. The S phase really this is the phase that we make more DNA. Because eventually, we are going to split, which means you now need double the DNA. And so in the S phase, the synthesis phase, we have new DNA being formed. And then in the G2 part of interphase, it's making sure now we've got double organelles, double mitochondria, double rough ERs, double everything. We're really getting as ready as we can before we go into that mitosis phase, because the mitosis phase can happen very, very quickly. And so it's the cell it's getting ready to divide, but it's not dividing yet. Now, for mitosis to happen, 1 cell becomes 2 cells. The cells that it makes, since we're just doubling our DNA, doubling our mitochondria, and I'm like, are you kidding? Since we're doubling everything we originally have, all of the offspring are clone cells. And I'm like, they're going to have the exact same information, exact same things that the cell before it had. Now, these, mitosis and cell division can happen continuously in some cells. Our skin and our intestinal lining are the biggest ones. Our cells are constantly reproducing. We're going to talk about skin in a little bit. But again, if I scratch my skin and I have skin cells, flying around, that's fine. And I'm like, because I'll get new ones, because I have so much mitosis just happening every day. And I'm like, in my skin and in my intestinal lining, we're constantly losing skin cells. So some cells are always undergoing mitosis. There's not a lot of lag time. Some cells don't like to do any mitosis. It's like what cells are there are the cells that are there. They just hang out in interphase all the time. Cells of the nervous system, your skeletal muscle and your cardiac muscle cells don't like to undergo mitosis. So if you damage these tissues, then what? Can you just make new? Shake your head. No. Once you damage these tissues, the cell can be like, oh, well, I've got a damaged cell, let's just make a new one of those. And I'm like, because it doesn't undergo mitosis, it's not going to repair. And so when you damage your nervous tissue, that's generally a permanent change unless, nerves get rerouted. But your original tissue is damaged. Same thing with skeletal muscle and your cardiac muscle. Damage to those cells and damage to those tissues is permanent damage. The best you get with a replacement is some fibrous tissue, but it's not going to do the same thing as nervous tissue or cardiac muscle tissue. Now when we want to divide our cell, and I'm like, we go through the mitotic division. Now, mitosis itself is division of the nucleus. So when I start mitosis, I have one nucleus. When I end mitosis, I have two nuclei. I've divided the nucleus itself. And there are the 4 phases, prophase, metaphase, anaphase, and telophase, for dividing the nucleus. So by the end of telophase, you have 2 nuclei. Then, when we want to split the cytoplasm, we have the cytokinesis. That's the full splitting in half. Did you guys get that right on your lab exam? Division. I think it was division of the cytoplasm? Yeah. Cytokinesis? And all of a sudden you're like, oh, no. Cytoplasm, cytokinesis, they both start with cyto. I looked at them like, Deke always asks one of them. Now, going into our 4 phases of mitosis, and again, we've talked about this in lab, so I'm not going to go over a ton of detail. The biggest things, your chromosomes become visible. You can see dark squiggly lines. Since we've doubled all of our chromosomes, that happened in interphase, the chromosomes pair up, attach at the centromere. Centromere was on the lab exam. They attach at the centromere. Now you have your big old chromosome. Centrosomes is the region where you find centrioles. They like to go hang out at the opposite ends of the slide, or the ends of the cell. I've got a picture. I'm just going to leave it up here. And I'm like, so your centrosomes, we've got our centrosomes with our centrioles. They like to go to opposite sides of the cell. Now, because in our lab, what did you have to learn what centrioles did? Do you remember what centrioles did? Yeah, I was going to say, they pull things apart. They help in cell division. And I'm like, and so if we want to split the cell apart, those centrioles have to go to the edges so they can do all of their splitting and pulling and yanking apart. Other things that happen, because I've got an early prophase here, and the next slide I have is a late prophase, is your nuclear envelope goes away by the end of prophase. So you have your nucleus, we have our chromosomes, they're attached to the centromere, our centrosomes with our centrioles are going to opposites, but I have one nucleus, but I need 2. So, and then I need to yank everything apart. And so our nuclear envelope by late prophase is gone. We have no nuclear envelope anymore. I have to yank it apart anyways. We just need to get rid of it. And I'm like, those centrioles inside of the big centrosome region, they send out little spindle fibers that attach right at the centromere. And I'm like, so those centrioles send out fibers that attach at the centromeres. And occasionally you start to see chromosomes heading to the middle of the cell in late prophase, like at the end of prophase. But what those spindle fibers are going to do when they're attached here, as they sit there and lengthen and shorten between one side and the other, they eventually get you into metaphase. When all those chromosomes, between the lengthening and shortening of the spindle fibers, all of those chromosomes are lined up right in the middle of the cell. They're lined up along an imaginary line called the metaphase plate. It's an imaginary line right in the middle of your cells. Once they line up, what happens to all those chromosomes? They get yanked apart. And I'm like, there's my imaginary line. They're all lined up on it. And I'm like, once they're lined up, they get yanked apart in anaphase because Anna's sad. And I'm like, they get yanked apart super fast. They just yanked. There you go, yanking. They get pulled apart. The sister chromatids get pulled apart from each other. And each chromatid, we now consider its own chromosome. So the chromatid becomes an adult, becomes its own chromosome. So they're getting yanked apart. One half the centromere went one way, the other half the centromere went the other way. sister chromatid said goodbye, and now we call them chromosomes. After anaphase comes telophase. Now, chromosomes stop moving. They've been pulled as far apart as they need to. The chromosomes themselves start to uncoil. They don't need to be these condensed chromosomes anymore. They kind of start to uncoil into that thin, stringy thing that you just look as, it looks like fuzz under a microscope. A nuclear membrane comes back, because we have to make a new nucleus by the end of telophase. The nucleoli that went away earlier in prophase, they come back. And any of the spindle fibers that went out to go attach to chromosomes, they go away. We don't need them anymore. A lot of what happens in telophase is the opposite of what happens in prophase. Because in prophase, nuclear membrane disappeared. Now it comes back. And I'm like, the spindle fibers here disappear. They appeared in prophase. So a lot of times prophase and telophase is kind of opposite of what's happening. Then to do a full split, splitting the cytoplasm, we have our cytokinesis. Because by the end of telophase, we have 2 new nuclei, all the number of chromosomes they need. Then we have our cytokinesis. Now, there are some microfilaments that start pulling and tugging on the plasma membrane. And as they start to pull and tug, they form, I think I've got a picture. I'm like, an indent in the cell. So as they start to pull and tug tighter and tighter, they start to get an indent on the cell. It's called a cleavage furrow. Yes, it's called cleavage. Just means an indent. Or it means, the official word for cleavage means to split into 2. So we get this cleavage furrow in the middle. And I'm like, you can start to see this. And it will sit there and pull tighter and tighter and tighter until the 2 cells are split in half. And so it's little filaments that just are pulling tighter and tighter and tighter, just like a rubber band would be just clamping down tighter and tighter and tighter until eventually it's 2 brand new cells. You've now split the cytoplasm in half. All the organelles have been split into half, 1 into each cell, and you have 2 new cells. Now, some things can happen with mitosis. One, we've got what's known as apoptosis and things that can modify cell division. There are times we need more cells in our body than we do right now. The biggest time is like during development, when you are growing taller, when you are just growing, because you have more cells in your body now than when you did when you were an infant. And I'm like, and so that development of growth, mitosis is happening faster and faster and faster. That's why kids need to eat more. And I'm like, because they are growing, they have mitosis happening all the time because they have to add all these new cells onto their body. And I'm like, so there are times that you need more cells than other times. And then there's other times that occasionally we just need to kill some cells off. And I'm like, we don't need them anymore. They're damaged, they're dying, we need to get rid of them. Occasionally some cells, and I'm like, if we want to kill them off, it's known as apoptosis. Apoptosis, its nickname is cell suicide. When the cell's like, I'm not needed anymore, let me just kill myself off. And I'm like, apoptosis. And so, yes, we start to have membranes of various organelles that start to leak. Your enzymes get out, they start to break down everything else. And as long as you can break down the DNA and your cytoskeleton, your cell dies. So you start to leak enzymes, and you die. Because occasionally you just don't need the cells anymore. So they'll program themselves to die. There's apoptosis. Now, some reviews. So pull out your colored note cards if they're not out already. And the first question, what must happen before a cell can begin mitosis? Pink, orange, yellow, or green? So before mitosis. There's things that happen during mitosis, but before mitosis. Now, and you can link all 3 of the 4 of them into one of the phases, and the other one, well, I guess all of them are part of the phase. And I'm like, it's pink. And I'm like, the chromosomes and DNA get duplicated in interphase. Sister chromatomes get separated in anaphase. The nuclear envelope breaks down in prophase. The nucleus divides into 2 by the end of telophase. So the orange, yellow, and green, those are all different phases of mitosis. But before mitosis can happen, we need to double our chromosomes. The centrosomes move away from each other, and the nuclear envelope breaks up during which phase of mitosis? So the centrosomes move away from each other, and the nuclear envelope breaks up. I was like, everyone has it. It is either orange or yellow, but it's only one of them, because half of you have yellow and half of you have orange. And everyone's like, oh, moving away. But what's moving away? It's orange. The centrosomes, the centrioles, move to opposite sides of the cells. We're not splitting the centromeres, and we're not splitting the chromosomes yet. And I'm like, just the centrosomes move away, part of the centrioles move away, and the nuclear envelope goes away in prophase. The chromosomes line up in the center of the cell during which phase of mitosis? You all got it right away. It is metaphase. They all met in the middle. The sister chromatids separate and begin moving towards opposite sides of the cell during which phase? I feel like I have these all in order. I need to switch these around. It is anaphase, which leaves us with this next one. The chromosomes arrive at the poles. The nuclear envelope comes back around during which phase? Yes, it's the telophase. And my last question, at the end of the mitotic phase, the cytoplasm divides in a process called? And it is green. Cytokinesis. Get rid of this. Where does it go?